Good afternoon, everyone. My name is Melanie Usher with the Arizona Telemedicine Program, the Southwest Telehealth Resource Center. Welcome to today's program, the Arizona Telemedicine Program Connected Health Webinar Series. So as you were connected, your microphone and your phone was muted. Please use the chat feature for any questions you have for our presenters. And we've also got our post-webinar evaluation that will pop up after the webinar is done. I will also be sending the link to my thank you email to everybody that you're used to getting, and you can link on from there. CME credit is available for attending the live presentation. And of course, all of our EMS folks, you'll respond to that email, and the groups that are meeting together, just go ahead and send me the names of all the folks in your rooms that are meeting together. This is being recorded and it will be listed on our website for viewing later. All right, so everybody got these in various emails. These are the outcome objectives, accreditation statement, relevant financial information for all of our presenters. I'm not going to read through this. Disclaimer, standard disclaimer that we have our presentations. And again, please look forward to next and upcoming webinars. When we get the next date and topic, I will send it out to everybody that's been attending. But here's our website where you can check for other programs that we have available. Come on in, folks. We've got some fellows coming in. And again, your opinion is valuable, so please go ahead and do that evaluation at the end. I'm going to turn it over to Dr. York, who will start with some of the introductions of our presenters today. Dr. York.
All right, so we've got a special guest today, Dr. Neil Ampel. I have known this guy at least the last 40, 50 years, it feels like at this point. So we work together, maybe more, I don't know. But we work together at the VA. This guy is everywhere. You've maybe seen his name pop up in any number of different presentations, publications, HIV, COVID, you name it, world expert on Valley Fever. And he is here today to talk about, I think, a really cool topic, not something that I think a lot of folks will be familiar with. And we've got Dr. Klotz, a man who needs no introduction at this point, who has got some other rather bizarre things to talk about that, again, I think not a lot of folks are going to be as familiar with, but I think you'll be very interested in some of these odd topics today. All right. Now, if somehow this is the first time you are joining us, I am glad you are here. But this is our webinar series on envenomations, intoxications, travel-related diseases. Now, if you are new to joining us, a lot of our past presentations are up on the website, the University of Arizona Telemedicine website. You can see past presentations. But a lot of our ideas are coming from you folks. So if there is something that it looks like we haven't covered, maybe you want to go more in depth with, please let us know in the evaluation at the end. We're honestly very interested in keeping this going until we're so sick of it, we can't do it anymore. So please keep giving us stuff that we can talk about. And we've got some of our topics here. I don't think we've covered quite a lot of these yet. We still have some real oddball ones to go, especially starting with today. I'm very excited for what we'll talk about, some sneaky topics today. But with that said, I'm going to turn it over to Dr. Ampel. You take as much time as you need. You've got the hour, sir.
Okay. All right, so I'm on.
Nope, let me get my
Hot mic.
Hot mic. Let me get my share screen.
Tell me when you're ready.
I was going to say, man, I thought you were reading off a teleprompter. We've done this so much at this point. She's got it down to a science, what she says.
I do, but
Impressive.
where's my share button here? There it is. All right, folks. If you remember from last month, my whole Zoom disappeared. We had some issues.
She's doing better.
Doing better. All right.
So we're sharing.
We are sharing. And I'm going to move this over here for you.
All right.
All right. And you are good to go. And you can use the.
Okay, well, I want to thank Steve and Larry and Melanie for inviting me. So today we're going to talk about tick-borne relapsing fever in Arizona. This was a case actually I saw when I was in infectious diseases clinic at Mayo Clinic. So, and it turned out that not only were there several cases there, but there were several cases down here in Tucson. And we put those together, and I think we should be aware of this disease in Arizona. So let's see. There we go. So this was the case that started this. It was a 72-year-old man who had developed shaking, chills, and fever back in June of 2017 while staying in a cabin near Greer, Arizona for the previous 3 weeks. He was seen initially at a local emergency department and was given amoxicillin for presumed sinusitis. Several hours after that first dose, he developed severe rigors and was admitted to that local hospital. At that time, he had a slight leukopenia of 1,800. Platelets were slightly down at 44,000. Bilirubin was up, AST was up, and his peripheral smear showed toxic granulations only. He was observed, he improved, and he was discharged. About a week later, he developed recurrent rigors and fatigue. At that point, he came into Mayo Clinic in Arizona ED, and again, lymphopenia was noted. He was started on fluconazole based on a positive EIA for coccidioidal serology, and again, his symptoms resolved, and that's how I saw him because he was referred to me for valley fever. You have to do that.
I guess I'll do it.
Yeah, I think you have to do that.
Okay.
Still not. Which one
There's
is?
the arrow.
Oh, we have to do it there.
There's-
Have to do it.
Okay.
Okay, got it. So 2 weeks later, his fevers again recurred. He then came again to the emergency department at Mayo Clinic, and he was given oral doxycycline 100 milligrams twice a day. Again, within hours of the first dose, he developed severe rigors, dizziness, and tachycardia, so he quit that. Subsequently, a test came back that had been ordered in the emergency room. It was a whole blood PCR for Borrelia miyamotoi, and that came back indeterminate. And because of this, the doxycycline was restarted because it was presumed he had some sort of spirochete disease. He completed that, he completed a 2-week course, and his symptoms resolved. Subsequently, immunofluorescence serology for Borrelia hermsii revealed a titer of 1 to 128 with a positive IgM and IgG that was done at CDC. Oops, this is very difficult. There we go. So, as I mentioned, we were able to put together a series of these cases and wrote them up in Emerging Infectious Diseases back in 2019 with the help not only of the clinical collaborators, but also of the state health department. Oops. So what we were able to find, in addition to my case, for 5 other cases that were noted both in Phoenix and Tucson between 2013 and 2018, all had visited cabins in the White Mountains in the late spring to early fall. In 5 patients, the first complaint was recurrent fever, and all were diagnosed either in medical centers in Phoenix or Tucson. Most had laboratory abnormalities, either leukopenia, thrombocytopenia, or elevated aminotransaminases, and all responded to doxycycline. And 2 had Yarish-Herxheimer reactions with antibiotic treatment. So this is a smear. This was actually taken by Brenton Roller, who is now a staff physician at the Tucson VA, but at the time was a fellow here at University of Arizona. So we use this in the publication, but this is classic. This is a right stain of just peripheral blood, not thick smear. And I think you can see the Borrelia spirochetes here, because the one thing about recurrent fever Borrelia is they are big enough to be stained on smear. I'm going to get this one of these days. All right, so let's talk about the history of relapsing fever in Arizona. So it turned out the first case was described in 1930 in a patient living near Greer. And I remember I'm in the basement of the medical school library, and I remember finding the paper upstairs when I was looking for it. So that was the first case, but most of the outbreaks, and there have been outbreaks in Arizona, have actually been up in Coconino County near the Grand Canyon. So in 1977, you can see 27 employees and 35 overnight guests were residing at, quote, rustic cabins on the north rim of the Grand Canyon. They found rodent nests in the walls and attics, and Ornithodorus hermsii ticks were isolated. So in 1990, there were 15 cases, again, overnight stay in cabins, not rodent-proof. And 2014, another outbreak among high school students who slept in a campsites-only cabin. that had been rodent-proof, but not mite-proof, and we'll come back to that. So these are the most recent outbreaks, and all of those were up near the Grand Canyon, not in the White Mountains. So just to look at these, we made this map that Haley Yaglin made at the State Health Department. So you can see the outbreaks up in Coconino County, and then you can see where our cases came from, way out in Apache County, up in the White Mountains. And I'm showing where those patients went. They either went to hospitals in Tucson or to Mayo Clinic in Phoenix. So the diagnosis was made in urban medical centers, not up in the White Mountains. And this is a picture I took when I was camping up near Greer. This is the West Fork of the Little Colorado near Mount Baldy. So it's very high, it's 8,800 feet. You'll remember there was just a big fire up there. And you can see the Little Colorado is really just a creek at this point. So maybe it is appropriate to call it the Little Colorado. But this is the kind of country where these cases were occurring. So this is a recent report from Center for Disease Control 2023, reporting all the tick-borne cases. So you can see most of them are intermountain. You can see up in Washington in the Cascades, down in the Sierra Nevada, the Rockies in northern Arizona. But there is one area, I got it this time. And you can see those are the ones in Texas. And these are actually cave-associated relapsing fever cases. So these are not rustic cabin or up high. These are low elevation cave, and that's probably Ornithodoros terracotta, a different tick and a different species of Borrelia. So let's talk about Borrelia associated with relapsing fever. So Borrelia are spirochetes. And all spirochetes have a relatively small genome. They're very, very thin, so they cannot be visualized routinely on gram stain. But Borrelia can be seen on Giemsa or right staining, Leptospira as well, but obviously treponeme's not. That's why we need special stains for treponemes. There are 4 forms. We usually say there are 2 forms, but I've expanded this. The first is louse-borne relapsing fever, which we don't see in the United States. And that, again, the vector there is louse. We see these in war-torn areas. I think the last case was either in Sudan or Djibouti. So not something we see in the United States. What we see in the US is tick-borne. And most of the cases are associated with Ornithodoros or soft-bodied ticks. And the ticks, there are multiple species and multiple species of Borrelia, of which Borrelia hermsii is the most frequent. But I do want to point out that there are other forms of relapsing fever. And the first is this Borrelia miyamotoi, which was first noted in Japan. This is associated with hard-bodied ticks. So typically, this is Ixodes. So this is very different in its geography because it's the same geography as Lyme disease. So there is a relapsing fever syndrome associated with Borrelia miyamotoi that is in the Northeast and upper Midwest, not here in the Mountain West. And recently, there's been a single case in Alabama associated with the Lone Star tick. So that's one case. So just to be aware of that. So I made this chart out to sort of divide this up of the major Borrelia. And one thing to point out to people, unless, like in our case, unless someone does specific serology or other very specific tests like PCR, you're not going to know what the Borrelia is. So often we're left not really knowing, making assumptions based on geography and site. As I said, Borrelia hermsii is the major organism. The vector, the soft-bodied tick, Ornithodoros hermsii, and this is Western US, as you saw, up into Canada, relatively high elevations, 1,500 to 8,000 feet. And this lives, and we'll come back why, in intermittently occupied cabins. You already got the sense of that. And the reservoirs are usually ground squirrels, tree squirrels, chipmunks. Now, Borrelia terracotta is the other one that's considered southeastern, That is a bigger soft-bodied tick, Ornithodorus turricata, lower elevations, and this is associated with caves. And again, the vectors are not fully defined, but are assumed to be prairie dogs, ground squirrels, rodents, cattle, snakes. And then finally, I mentioned Borrelia parkeri because I know Steve is going to talk about this, so I'm stealing his thunder. But actually, the cases I found, so I don't know what he's going to say, so I'm holding my breath because these are really rare cases. There's one confirmed case in Colorado, has very similar to Hermsii where it lives, and the ground squirrels considered its reservoir. So I made this, well, I didn't make this chart, I colored this chart. This is from MMWR, so to go over the symptoms. And you can see 97% of the symptoms, these are the cases reported to CDC that they published in 23, have fever. And then you see, about 2/3 of headache, myalgia, and chills. So the main thing is this recurrent fever. And that was the issue with my patient. He kept, he came back 3 times with recurrent fever. So you have to have that in your thoughts if you're practicing in a region where relapsing fever may occur. Then you see these other symptoms that are fewer than half, but still not insignificant: nausea, vomiting, sweats, fatigue, malaise, anorexia. And then the rest are uncommon. But I do want to mention neurological or ocular symptoms because one of our cases had uveitis. So you can see ocular disease from this. So again, what are the symptoms that one sees, or what are the laboratory abnormalities? And that's the other key thing. It's not, and that should have really tripped us off on this patient's first visit, right? Because he's having fever and he's got lab abnormalities. And it certainly should have tripped them off the second time when they kept seeing leukopenia and elevated aminotransaminases, because you have those two things and you should want to put it together. It's a good thing. It's a good advertisement for maybe having clinical AI, because I think if you ran this through AI, recurrent fevers and elevated LFTs, and I think AI, because that's often what happens, and it's what happened to me. He saw me for valley fever, so I was not thinking tick-borne relapsing fever. So these are the things: thrombocytopenia, both leukocytosis and leukopenia. You can get a mild to moderate anemia. Again, elevated aminotransaminases, elevated bilirubin, like my case had, elevated ESR, and even coagulation abnormalities. So that combination is really the key. So how do you diagnose this? Well, like Brenton, the fastest way is microscopy. And that really, if you can get the lab to do a smear, and you can, if you can work with your lab, you can do thick smears, but just doing a right stain when the patient's febrile, that has about a 70% hit rate. But obviously you can miss. You can do serology, as we did, but only certain labs do. In this case, CDC did it. So that's only retrospective. You're only going to make the diagnosis weeks later when you see these fourfold rises. PCR can be very helpful, but you have to have a lab that can do that. Culture is rarely done, and obviously, I think nobody does animal inoculation anymore. So the key is to ask the lab to do a right stain and to look at it with the pathologist. So here again, do a right or Giemsa stain. You can get acute or convalescent serology. Remember, Lyme disease serology, because that's a Borrelia, may be positive, and VDRL may be positive because Borrelia are spirochetes, and so is our treponines. And remember, when you do this, like what we had to do, you have to work through the state health department. And remember how the health department works. So if you're in Pima County, you've got to go to Pima County Health Department. If you're in Maricopa County, you've got to go to Maricopa County first. It goes to the state or to the county health department is who you report to. You don't report to the state. Then they go to the state. So it is a kind of a convoluted process if you think you have a case to make the diagnosis. So go to your local health department, say, I think I have a case, and they'll go ahead and work with you getting things. And in fact, they they constrain your testing. We couldn't test on our own. And again, how we picked this up was someone was able to do a PCR at Mayo Rochester for Borrelia miyamotoi, and it just made people think about it. Treatment is doxycycline, 100 milligrams BID for 10 days. For CNS disease, which is uncommon but can occur, then we would use either penicillin G or ceftriaxone. So why do Borrelia do this? Borrelia have this interesting antigenic variation. They have genes for serospecific outer membrane surface lipoproteins on extra chromosomal linear plasmids. And these are usually quiet. But if the gene recombines at another site on the plasmid, it may become active if it's downstream from a promoter gene that gets activated. And so, for example, Borrelia hermsii has 30 of these variant genes. So technically, you could have 30 episodes of fever, because what's happening is the Borrelia is presenting to you different antigens at every cycle. So you clear it, and then boom, this thing cycles through, presents new antigens, and that's why you have these recurrent fevers, usually about a week apart. So you get these waves of distinct organisms, an immune response, until you burn out. So what's the vector? The soft body ticks, there is Ornithodorus hermsii. So these are, and Steve and I were having fun with this, nidicolous. We think that's how I say it. So not ridiculous, but nidicolous. So the way to think of these, if you've ever run into hard-bodied or exotic ticks, they live in the woods. They live close to the ground on grass. I was hiking on an island, Lewis Island in Scotland. I got a questing tick, and that's where I was. I was in grassland. So that's where they live. These don't live there. They live with the rodent burrows, and then that's where they live. So if you have a cabin that's become infested, then it can be secondarily infested by these soft-bodied ticks. And there's the rustic cabins, and this was the joke because this was presented at Portal, and that was my cabin. I was in the rustic cabin, so I was pretty nervous the whole time I was there testing. I think that cabin reportedly was filled with reduvids in the past. So wow. Yeah, that's the story on that cabin. But also in caves, as we said. So the difference with these are living in close proximity with what they feed on. But say you have a cabin and you go and clean it out. You get rid of all the pack rats. You get rid of all the rodents. The ticks are still there, and that's why you saw all those outbreaks up in Coconino, and that's what happened in Greer. This guy hadn't used that cabin in a long time. So the ticks are there, and the other thing about these ticks is they feed at night 20 minutes, and they have a fairly minimally symptomatic bite. So you don't notice being bitten. And then the ticks are very persistent. So they only need a blood meal every 5 years. So they're just waiting for you. So the big key is here, you not only need to rodent-proof your cabin, you need to use acaricides or anti-tick, anti-mite. And if you don't do that, those ticks are still there. So think about that whenever you go to these rustic areas. There's a place in Hannigan Meadows that I'm sure I should have gotten tick-borne relapsing fever. I won't mention the name, but I think there's only one place up there. So I'll probably get in trouble. So the final thing is, you notice this patient got amoxicillin and doxycycline and really got sick after that. And all infectious disease doctors know this is the Yarish-Herxheimer reaction. So this is a classic reaction we see with secondary syphilis. So when we diagnose syphilis, then we give the patient penicillin, and then within a few hours, they're back with fever, chills, hypotension. So we know whenever we treat a patient for syphilis, particularly secondary syphilis, we have to warn them about a Yarish-Herxheimer reaction. This has gotten into the parlance for recurrent Lyme disease and so-called chronic Lyme disease. So people who believe in the And they've sort of balderized it to a Herxheimer reaction. It's really interesting because we used to see these patients and we'd know that they had been informed, however incorrectly, because they said, and then I got the Herxheimer reaction. So all these spirochetes you can get when you treat, and the presumption is you're releasing exogenous endotoxins into the system. So you always want to be aware of that, and you want to be aware of it in tick-borne relapsing fever. So I think that is my last slide. So I'll end there.
All right, do we have any questions? But go ahead and put them in the chat. Looks like we've got a couple over there.
Yeah, we got a fair, all right, so when the Borrelia recombine to present different antigens, they must have been incompletely cleared from the body. Where does the spirochete hide, so to speak, from the immune system in between relapses?
You know, that's a great question because all spirochetes, including syphilis, have very, very long half-life. So they tend to live in endothelial tissue, and they're almost always incompletely cleared. And that's an issue with syphilis, and that's why people get tertiary syphilis. So they live very quietly, they're very slow growing, probably in endothelial and vascular tissue. Again, if we take the syphilis model, when you get tertiary syphilis, that's all vascular endothelial damage. So they're probably just hanging out there. My presumption would be, yeah, they are incompletely cleared. Eventually, the immune system, I think, for these Borrelia will clear them. But it takes those multiple episodes.
All right, and then does Rocky Mountain fever have this?
Well, Rocky Mountain fever is a rickettsia. So it's a different organism. Yes, we would treat with doxycycline, but it doesn't have recurrent fevers. No. So in fact, as you probably all know, Rocky Mountain fever, which we do have, particularly up in the White Mountains. So maybe we should call it White Mountain fever now, because it was particularly prevalent. There was a large outbreak on the reservation there. So that you get very ill. Obviously, you have the rash, an ascending rash, and very high mortality. So you're not going to confuse. That's a very acute disease. It's similar in that patients are going to present with headache, rash, fever. So the rash would be different, and they're going to have all the same laboratory abnormalities. And the treatment is the same. It's doxycycline. And in fact, what we know, and I'll just give you an anecdote, while the original outbreak was going on in the White Mountains, so the VA, when I was there, was a transfer site for the Indian Health Service. So we got a patient down. The house staff came to me and said, Dr. Impel, they think this dialysis patient who had a fever on dialysis has Rocky Mountain spotted fever. And he had no rash. And I'm thinking, this is kind of ridiculous, not ridiculous, but ridiculous. And in fact, someone from CDC called me quite late for them and said, no, Neil, you got to treat this guy because you can have mortalities of 70% from Rocky Mountain spotted fever. So the patient ended up not having Rocky Mountain spotted fever. But during this outbreak, people were being empirically treated because they had had too many deaths. So there are similarities, but again, Rocky Mountain is a rickettsia. One episode, you will see the same lab abnormalities, but you will see the two things are headache and that rash. So a little bit different. Do the squirrels and mice get ill? I have no idea. I don't know if this is endozoic or they're sick. And in fact, I think the host, and Steve maybe wants to talk about this, are not very well defined for these organisms. I don't know of any link to, the question is there a link to lupus erythematosus? I don't know of any link of relapsing fever to lupus, so not that I know.
I have a question. Would these, would it affect older adults or people with other comorbidities more than say a young
I don't think we
person?
know. These are so sporadic. And the first case was that first 1930s case in Greer was a fairly young man who'd been living up there. So no, I don't think we know enough. It's possible if you're older, maybe you wouldn't do as well. But I would say not only that guy, but the high school students got pretty sick as well. So probably not, no.
Okay. And one piece of information, 5-year feeding cycle, they only feed once.
No, they'll feed, well, no, if you're sitting in, you know, in the rustic cabin, they're gonna feed on you all the time. But they can they can live without eating for 5 years.
Wow.
And they do that without Ozempic.
And they can live up to 20 years.
Yeah,
Actual life
so
cycle.
that's really the problem. So if you don't do both, and that's very common, and we all know that because we all have pack rats here, right? So when you get rodent infestation, you think, well, and I think that's what happened with my patient. He thought he had cleared them out, but and of course, cases in the White Mountains hadn't been reported. That's why we reported this. So you wouldn't be thinking of relapsing fever up there. So you thought you were fine. You got rid of the rodents, but in fact, the ticks were still there.
So would you say that our EMS folks that might be, you know, reaching out and they're out in the rural areas, other information such as the background, where maybe they've been traveling within the past few weeks, or what other information would be helpful to get before they go?
Well, this case, because when we looked at them, they're all from around Greer, which is, or that area. So absolutely, if you know it's there, then and that was very important in our case, because this was a guy from Phoenix. So I sort of talk, oh, he was up in his cabin, but that took time to ask. Of course, he initially presented, and I'm assuming he went to Springerville. That's my recollection. So that would be really important for the ER in Springerville to be aware if people came in with multiple episodes of fever.
And how quickly would it present if someone got bit?
It's
They're up in their cabin.
About a week.
About a week, okay, this week. All right, do we have any other questions? Oh, we got a comment. The chief had Rocky Mountain fever, now has lupus.
Yeah, so don't know if that's related.
Okay. All right, any other questions for our soft body ticks? And then we will turn it over to Dr. Klotz, who's going to be talking about hard body ticks. Dr. Klotz, you want to go ahead and pull your slides up? You're muted.
Oh, still muted.
All right. Here comes Dr. Klotz. All right, Dr. Klotz, take it over.
Okay, thank you, Melanie. Great talk, Neil, and really pretty entertaining, I'll have to say, as usual, and full of good facts. Good afternoon to everyone, and thank you for joining us. I've got about 15 minutes I want to spend just talking about some hard body ticks. I've entitled this presentation, Some Exotic Ticks of the Southwest Gaining Notoriety, meaning hard body ticks. And I think, first of all, Neil covered very nicely the difference between the two ticks, but I'll just point out a couple features here that I think are important. As Neil said, the argacid ticks or soft body ticks are really long-lived, and furthermore, they have multiple nymphal stages. I know one tick that we worked with that caused an outbreak of biting humans and other problems in one of our hospitals locally in Tucson had as many as 11 different nymphal stages reported, and it requires a blood meal between each nymphal stage to occur. Whereas hard body ticks, the ones that you can see the mouth parts on when you look at the dorsal aspect of that tick, they're very slow moving. And as opposed to the soft body ticks, you can't see the mouth parts. As Neil's photo showed, all you can see are the legs underneath them. And they can move a little faster. But the hard body ticks will lay up to 3,000, maybe 8,000 eggs at a time. And this is in a nest, which has led to Neil's ridiculous or nediculous statement, which means nesting. Whereas the soft body ticks the females never provide more than 500 eggs in a lifetime. and there would be just a few of them after each laying of eggs. And other things Neil covered very nicely between the two different families. So I want to talk about two exotic species for today, Dermacentor occidentalis, which is the Pacific Coast fever tick, which transmits Rickettsia philippi, and Amblyomma maculatum complex, which has been recently reported in southeast Arizona. It causes a tick fever, and it transmits the pathogen Rickettsia parkeri. And just when I was preparing this, I remembered that Didier Rouault, whom you may remember him, he was the gentleman who sort of tried to use or proposed using chloroquine for the treatment of COVID-19. And now kind of lives in infamy because of his behavior and these claims. He actually discovered the difference between Rocky Mountain spotted fever in a patient versus that caused by Rickettsia parkeri, the organism we're talking about, which is now in southeast Arizona. It's part of the Rickettsia spotted fever group. And this group is a bunch of different Rickettsia, is usually approached by ordering a serology. The serologies are helpful, but they're not very specific, and there's a lot of cross-reactivity. And it took Didier Rouault to provide a PCR to distinguish RMSF, Rocky Mountain Spotted Fever, from the Rickettsia of the tick fever, which we're talking about. So tick fever caused by Rickettsia parkeri, the pathogen was first described back in 1937 by Parker. The first human case in the United States was reported in 2002 in Virginia. Nevertheless, this is the second most prevalent spotted fever next to Rocky Mountain spotted fever that is found in the United States, and it's found throughout the Western Hemisphere. But the vectors in South America are entirely different. They don't belong to this Amblyomma maculatum complex. The feature of this disease is that eschars are seen with it. They occur at the site of the bite. So an eschar is really just the vascular infarction, probably from the rickettsia proliferating within the endothelial cells, causing a little petechial lesion, which leads to necrosis. They aren't very large, and they aren't very large in the next tick we're going to talk about. But eschars, well, you're all familiar with them. You've seen bed sores. This is really an eschar caused by pressure, but they can be quite large. And I remember in one case worrying about this being Bacillus anthracis rather than a rickettsial disease, which I'll explain a little later, and in which case there's a lot of edema in the tissue and a lot of death of tissue. So these can be large, leathery scabs that persist for weeks and months. But we're talking about eschars that are almost pinpoint. So this is the tick fever, the one that's found down by Portal, Arizona, and Rodeo, and caused disease in several people in the last couple years, some of whom have attended our conference down there that we have annually. And it's the tick is Amblyomma maculatum. You will know amblyomma from Amblyomma americanum, which is the Lone Star tick that is implicated in many other infectious diseases. But here are some of the rash that is seen after the bite. Oops, excuse me. And this particular lesion in D will go on to cause an eschar. In other words, it'll turn black and eventually fall out, leaving ulcerated tissue. So the Pacific Coast tick fever, which is caused by Rickettsia philippi, this is something of a little more recent origin. It's transmitted by Dermacentor occidentalis, and the Pacific Coast tick is found predominantly in the chaparral that is along the Pacific, all the way from Oregon to Baja California. And it too, its bite, if they carry Rickettsia philippi, will lead to multiple eschars, not one, but multiple eschars, wherever they're biting. And as Neil has pointed out, these are all easily treated with doxycycline, just 100 milligrams BID for 10 days or so. And the specific diagnosis can be provided by PCR, which can be sent off to the CDC. This has reached sort of troublesome levels in the Pacific Coast in the recent couple years. So that's the area along the Pacific that's the ticks reside, Dermacentor occidentalis. Here is that particular tick. The female's on the left with a very small scutellum or scutum, and the male is here on the right, and the scutum takes up almost all of the dorsal aspect of that tick. And of course, the female is the one who gets so large, you just see a little white tissue at the very behind the head here when they're feeding on humans. That is an adult. So, as Neil tried to point out, we're going to encounter spotted fevers, and one of which we've had for a number of years now, related to the brown dog tick, is Rocky Mountain spotted fever, which has been endemic in some of the Native American reservations. It's normally feeding on dogs. Dogs will die of this disease, but they normally don't. It's caused by Rickettsia rickettsii. These are protobacteria, so they're not ones you can stain with Reich Giemsa and see them, nor can you culture them. And its RMSF is found from the USA all the way to South America. And in the United States, the tick that carries this disease is both, as you can see, 3 different species, or genera, Dermacentor, Rhipicephalus, and Amblyomma. It's a moderate to severe infection, as Neil pointed out. There can be 70% lethality if not recognized and treated. It's easily treated if it's treated early. And unfortunately, the rash, particularly in the epidemics we've had in Arizona, hasn't always been present up to about 20 or 30% of the cases. So you need to start when the fever begins, and you need the right epidemiological picture, like Neil was pointing out. Another tick, or another rickettsia, and I never saw the tick in this case, was that I've encountered in travel clinic, was Rickettsia africae, which usually occurs in individuals in our months of November and December, but it's happening in another continent where people are going on safari and out in the bush in sub-Saharan Africa, where they're fed upon by Amblyomma ticks, which carry the infection, Rickettsia africae. It also has one or many different eschars that are a tip-off to this disease. And this was one that I saw 2 fellows with this who came to clinic. They had just returned from South Africa, and they'd been out on safari. And one fellow had a very large well, I thought it was large, eschar, large enough that I began to worry about Bacillus anthracis. But it was undoubtedly African tick typhus, and some doxycycline solved the problem. More spotted fevers that we see in the United States and has this sort of similar epidemiology would be rickettsial pox. And this is caused by the bite of actually a mite. I'll show you a picture of it later. But normally they're feeding on house mice. So this particular disease generally occurs in heavily populated areas, poor areas. Got to have a lot of mice in your house. And it usually follows eradicating the rodent, leaving nothing for Liponoisoides sanguineus to feed upon. So they begin to feed on humans. And their bite's going to lead to an eschar. And this is a tip-off to the problem. And there is also a flea-borne spotted fever that is found mainly in Hawaii the last time I looked. But this is caused by the cat flea, Encephalides felis, which will bite, again, if you've removed your cat or something like that, and the fleas are in the house, they will bite you. And if the cat has the rickettsia and the fleas have fed on it, they'll transmit this to a human. And leads to a rash and also an eschar. So the rickettsial diseases as a group, as Neil pointed out, or many of these tick-borne diseases, rickettsia in particular feature a fever, severe headache, which is very typical of RMSF, myalgias, muscle pain, and a rash if you see it. But as I pointed out, in the case of the RMSF, that particularly in the Apache Reservation, the breakout there, the number of patients with rash was not very high. And so the recommendations came down, if you find someone who's from an epidemiologically relevant area, has a fever that's unexplained, you just begin them on doxycycline. And we've been doing this for years at the hospital. The rash typically develops 3 to 6 days after the onset of illness. It often starts on the patient's limbs and then spreads to the trunk and may be seen on the palms and soles of feet. It is typically maculopapular. Petechiae may occur, and these can lead to areas of necrosis. So people can end up with amputations with this disease because rickettsia are targeting the endothelial cell. And cutaneous eschar may be seen at the site of a ticker bite or mite bite in some of the rickettsial diseases, and typically develops 4 to 10 days after this inoculation of the rickettsia. So here are some 3 interesting vectors. Here's the cat flea down here on the lower left. This is Encephalides felis, and it transmits Rickettsia acari, particularly in Hawaii. But here is the one that transmits rickettsial pox. This is a mite, which looks superficially like a tick, but these are soft-bodied animals. They're very small. This is probably a little less than a millimeter in size. You'll need a microscope to look at them. And if you're familiar with chiggers in the Midwest, this is what this is kind of related to, this mite, Liponoisoides sanguineus. And I just threw in this other mite to show you some size distribution here. This is the one that you'll see in Arizona, usually in the monsoon months. After a rain, they come out once a year in large numbers, and they sort of drift around on the rocks on a surface, hundreds of them, thousands of them. And no one predates them. No birds will come down and eat them because they taste so very, very nasty. But they're very plump, they're very beautiful, And the life cycle of this particular mite was very vividly described to all of us at one meeting we had in Portal by our former collaborator, Justin Smith. So I'll end it here, and if there are any questions, I'll be glad to answer them.
There you go.
So Steve, this is Neil. So just to point out for flea-borne, there's a nice New England Journal paper on someone who went to a ranch in Texas. And then I think there was an MMWR, so there was almost an outbreak of flea-borne disease in Los Angeles. So it is seen in California and Texas in addition to Hawaii. So just so people are aware.
Yeah, it's pretty widespread. The flea is worldwide in distribution. And of course, it's integral to cat scratch disease.
All right, no other questions yet. We were talking about it here. So Justin Schmidt, you had mentioned, wasn't his last presentation, he talked about these velvet mites, didn't he? He had taste tested them.
Yeah, I forgot to mention that it's
I think
velvet
this was,
mite.
he said this was the worst tasting thing he had ever tried.
That's right.
That was my takeaway from that presentation.
So that would be another Justin Schmidt scale. He has the stinging scale, and now he can have the taste scale. But we actually saw them on the David Yetman Trail after a rain, up in the Tucson Mountains. My wife, because they're very, they're so red. Yeah, they really stand out.
Amazing. Yeah, and Justin actually, the unique part about him was not only being stung by things, but he ate all the things that he couldn't figure out why they weren't predated by birds and things. So, for example, we wrote a paper, it was in, I don't know, some ornithology journal, I've forgotten what it was. But he had a western kingbird outside his window in his house that was feeding on all the bees that were coming from his hive, but he noticed that they were all drones that the bird ate. It didn't eat the females, the workers, because they had stingers. So he tasted the workers, the worker bees, and they had this kind of bitter taste because of the toxin in their poison sac. And whereas the drone bees, he said, tasted like custard. They were just delicious. So it's just strange things like that always occurred with him.
This reminds me when Dr. York was talking about people licking Colorado bullfrogs for the toxicity.
My dog was.
I can't think of anyone that would go looking for these to give them a try. We've talked it up enough at this point. Now I'm a little curious myself.
Well, that's it, Larry, from my end.
All right, we've got a few more minutes, so if y'all have any more questions, do put them in the chat here.
Don't see any new ones. Again, the evaluation for today's program, it's going to pop up behind the screen once the Zoom is closed. Otherwise, I will be sending out the URL link in my thank you email for everybody that attended today. And again, please, in that evaluation, give us some more ideas. Like I said, Dr. York was talking about, they do take the ideas from you folks that are watching of what else they might be able to entertain you with. All this information and education, right? So let's see, we've got a.
I like the comments.
So comment we have,
Fair.
thank you, it's lots of interesting knowledge, mildly disturbing, but interesting. Thank you again, yes.
Same, much the same.
All right, everybody, so without any further ado, we're gonna go ahead and end it. You'll get 6 minutes back of your day. 5 minutes back, it's just turned. And please look forward to my email and our next session. I don't think we have our topic yet, but I'll be sending out that information as soon as it is scheduled. So again, thank you everybody for joining us, and we'll see you next month.